introduce our speakers for today. Aislinn Prendergast is currently the area lead for the Quality and Process Improvement team at Melbourne Water, managing a team of process engineers responsible for providing technical support to the operational field teams. And the team is also responsible for ensuring the safety of the water supplied to customers through a risk-based catchment-to-tap approach. This includes monitoring catchment controls, treatment performance, and downstream distribution and storage. Previously part of Melbourne Water's asset management team, responsible for raising and overseeing minor and major upgrade projects. Our second speaker is Dr. Brett Bruin, and he's been working in the field of microbiology for over 3 decades and has 25 years experience in the analysis of water and the presence of waterborne pathogens. He's worked closely with labs and experts around the world developing new technologies, training analysts and establishing best practice in order to optimize complex technical procedures and to generate robust methodologies. Now, you'll have a chance to hear from Aislinn a little bit later in the webinar, but now I'd like to turn it over to our first speaker, Dr. Brett Bruin. Brett?
Thank you, Dean, and welcome everybody to this presentation. I will be covering some of the more general aspects of the analysis of water for the presence of Cryptosporidium and Giardia. For those of you who are not aware, Cryptosporidium and Giardia are very, very common protozoan parasites that can cause gastrointestinal illness. These illnesses are typically self-limiting, but in certain cases, they can be fatal. And it's in the usual groups, so the very young, the very old, people who are immunocompromised in some way. Where do we find them? We've already indicated that they are pretty much ubiquitous. So anywhere that you're going to find water, particularly within the environment, you're going to find Cryptosporidium and Giardia. So rivers, lakes, the sea, swimming pools are another potential reservoir for Cryptosporidium. And also you can find them in drinking water. And we've had incidences in the UK where water utilities have found Cryptosporidium in the drinking water supply, and they've had to take a lot of remediation, and it's quite costly when outbreaks do occur. Just to give you a little reminder about routine water monitoring, it is a verification process. So we're doing the routine monitoring to verify that everything from the source water to the finished product is done correctly. Verifies water treatment is therefore effective. We usually test for fecal indicators. However, even though those fecal indicators can indicate a pathogen risk, there are some exceptions. If there are no indicators, we do have an effective treatment and a low risk of pathogens, but there are some cases where that's not the case. And this is the caveat that we have. So if we look at bacteria we're commonly looking for in the water supply, E. coli, salmonella, chlorination, chloramination, ozonation is typically effective. Looking at viruses, as long as there's sufficient contact time, again, typical water treatment practices are effective. When we get to some of the sporulating bacteria, Clostridium and Bacillus, water treatment becomes less effective. We're looking at more physical barriers to try and remove that. And then when we get to Giardia cysts, and particularly Cryptosporidium oocysts, the typical water treatment processes are becoming ineffective. So we would then, in that case, look for those pathogens specifically. Everything pretty much started back in 1993 with a very significant outbreak of cryptosporidiosis in Milwaukee. Very interestingly, the pharmacies actually made some money out of this because they were stocking Imodium. But nearly 900 people were put on a boil water notice, which is the only effective treatment once Cryptosporidium gets into drinking water. There are probably over 400,000 cases of gastrointestinal illness, and that's an estimate because a lot of people would not report this. There were nearly 70 fatalities, and close to 100 million in associated healthcare costs. So it can be a significant risk if there are outbreaks. How do we prevent outbreaks? Over the years, we've improved water treatment, particularly with ultrafiltration and UV treatment, which are very effective as either physical barriers to Cryptosporidium entering drinking water, or the UV treatment would effectively render the Cryptosporidium and the Giardia non-infective. But also, we should be doing routine monitoring, both pre and post treatment, and that's very, very important. So we know what's in the influent water, but also what is also in the finished product. We're therefore assessing again the efficacy of water treatment. There is a challenge. Analysis is always quantitative. One thing to remember about both Cryptosporidium and Giardia is they're not like bacteria. They will not reproduce outside of a host. So we can't use our typical laboratory type tests to grow the bacteria and then assess the number within a sample. Limits are very low, typically less than 1 per 10 litres of water. And then we're looking at taking a grab sample of water. For raw waters, we would typically look at 10 litres. For drinking waters, much larger volumes are required, up to 1,000 plus litres of water. But then we're going to go through a process and we're going to concentrate all of the contents of that water sample onto a 9mm microscope slide. So it's quite a large challenge. How do we go through that? The methodology is very, very complex. It's very time consuming and requires a lot of experience. We go through sample collection. So this usually involves some form of physical collection using filtration media. Then we go through elution. Here I'm looking at a Filta Max Xpress elution station. We'd then go through some more concentration steps, and we'll cover that in a little bit more detail shortly. Then we'd look at purification. Obviously, if you're physically concentrating the Cryptosporidium and Giardia, you're also physically concentrating a lot of particulate matter. We have to separate out the target from all of that extraneous material. And then we go through a process of visualization. And again, this is probably one of the hardest parts, requiring the most technique. Enumeration is via microscopy. It can be very, very subjective. And labs often collaborate when there's an outbreak to ensure that they are correctly identifying organisms. IDEX supply means for sample collection. So in terms of filter modules, we also supply equipment to perform the elution process. And we also supply the necessary products for purification steps. So just covering things in a little bit more detail. If we look at a typical sampling rig, we'd have an inlet, probably with a pump in case of low pressure events. And then we have the filter module itself and a number of pieces of equipment such as flow totalizers, flow restrictors, flow meters, so that everything is done in a reproducible manner. For elution, there are two options that IDEX supply. One is a manual elution station. We typically recommend that for low volume users. It's quite labor intensive, taking about 20 to 30 minutes to complete per sample. For more high volume users, we have the Filta Max Xpress. This is automated, and elution is consistently done in 2 minutes. For sample concentration, again, there are a couple of options. If you're using the Filta Max only, there are options for membrane filtration to concentrate that sample. But then additionally, it needs to be further concentrated. So for Filta Max and Filta Max Xpress, we also include a centrifugation step where we're concentrating all of that material, both targets, Cryptosporidium and Giardia, but also the particulate matter, into a small pellet at the bottom of a centrifuge tube. The IMS step is quite labor intensive and is one of the most important parts of the technique. In summary, we're taking our concentrated material containing our target organisms and all of the particulates, and mixing it with some buffer and paramagnetic beads. And then we're going to incubate that for approximately 1 hour. And then we're going to use a magnet to pull those paramagnetic beads out of the sample with the target organisms, Cryptosporidium and Giardia. And then we'll go through some cleaning steps and do a further concentration. Ultimately, we're ending up going from a 10 milliliter sample down to a 50 microliter sample. And then we can split the paramagnetic beads from the target organisms. We have to do that, otherwise they would just completely coat the slide and you would see nothing. So we're adding a buffer and then performing a neutralization step and then applying our final sample to this 9 millimeter microscope slide. So we might have gone from a 10 litre to a 1,000 litre water sample all the way through this process down to a 9 millimeter slide. Covering the way we identify these organisms, it's very, very subjective. We try and improve identification methods using specific fluorescent stains such as FITC or FITC. We also use non-specific nucleic acid stains. The one that's typically used is DAPI. And then we also use optical techniques such as differential interference contrast microscopy. And then the identification, again, very subjective, but it's based on morphology, so the shape of the organism itself, the intensity of the FITC and the DAPI staining, and looking for internal structures using DIC microscopy. One thing I would just mention at this point with regards to DAPI is it can cause problems because it's non-specific. It can stain nucleic acid within plant material. It can stain DNA within algae. And so in certain situations, it might actually make the identification more difficult. So we have this plethora of techniques to try and subjectively identify these organisms. What do we look for? With the FITC staining, we're looking for this typical apple green fluorescence. We get a bright halo. We're looking for typical size of the organism itself. And then with DAPI, you'll find up to 4 stained nuclei within a noocyst. Sometimes they get broken open, so that makes things a little more difficult. But these are the typical structures we're looking for. And then also in certain situations, particularly at higher magnifications, like 100 times magnification, we can identify sporozoites within the oocyst itself. With Giardia, it's a little easier because it's a slightly larger organism, but again, we're looking for this bright apple green fluorescence and this typical halo. And then we're looking for the nuclei within the trophozoites within the cyst itself. And then there are a number of different internal structures within Giardia that can be clearly identified. Nuclei particularly, the cytoplasm and the cell wall. Medium bodies are a little bit more difficult, but with a lot of experience, you can identify these internal structures and make that judgment call. When we're looking at method expectations, we try and do a lot of training, and we usually do training in reverse. So we'd start with staining, and then we'd work through the IMS, then we'd work through centrifugation, and we work backwards until we've got the whole method in place. For staining controls, we're looking for 95% ideally. For IMS spikes, we're looking for 85% plus recoveries. For buffer spikes, where we would go through the centrifugation step, 70% plus. With quality control samples, 50% plus. And again, for matrix spikes and PT samples, we're really looking for 50% plus recoveries overall. These are our internal guidelines. There may be different guidelines within, for example, US EPA method 1623. There are a number of regulations and guidelines. We just mentioned 1623.1. Testing is now regulated by the state, and there are no US-wide guidelines in place. The UK Drinking Water Protectorate had a very, very highly regulated guideline in place. This was repealed in 2007, and testing is now regulated by the utility based on risk assessments of their influent water. In France, the Department of Health has NFT 9455, and they only test what they would consider defective water systems. PUTAG is the Pool Water Treatment Advisory Group. There's no routine monitoring of pools, although there have been some studies that I'd be happy to share with you that have shown where analysis of pool waters has been performed. We do find Cryptosporidium and Giardia, even though treatment within the pool itself is deemed to be acceptable. World Health Organization recommends to assess systems to ensure proper function, and that's done using a water safety plan. And then with the EU drinking water directive, it's all risk-based. There's no mandatory testing. So it's very, very different around the world. The region where the majority of testing is performed would be within the United Kingdom. Unfortunately, even though routine monitoring is ongoing, outbreaks are still occurring. There are Cryptosporidium outbreaks within swimming pools. Animal feces from animal carcasses is still entering the water system, and we do get outbreaks caused by Cryptosporidium. And from 2009 to 2017, this has increased significantly. The reason why we're still having outbreaks, defective water treatment, aging infrastructure, the very complex routine monitoring. It can be quite expensive to put in place all of the equipment and to have analysts trained to perform this very complex technique. So expense, resource, time, and technique are probably the four things that restrict people from doing increased testing for Cryptosporidium and Giardia. What do we at IDEX try and do about that? Well, one of the things we cover is training. Training is absolutely key to performing this method, and constant monitoring will improve training over time. So we have a number of analysts. These are some of my colleagues from the UK that have helped us perform training over the years. And we've had numerous groups from around the world come through our laboratory to learn the technique and then apply it within their own laboratories. So in summary, Crypto and Giardia still do pose a risk to public health. Water treatment can fail. Routine monitoring is, in my opinion, essential. The techniques are complex. We appreciate that very, very much. But we continue to invest in it. We continue to try and develop the best technologies for this analysis, and we invest in the training of our customers. I just want to go through one more thing, which is it's not really a case study, but it's just to outline why it is important to monitor. So back in 1997, there was this very, very strict regulation introduced in the United Kingdom. This required utilities to test for Cryptosporidium at what were deemed risk sites. And it was 1,000 litres of drinking water every 24 hours, 365 days per year. And the maximum allowable limit, 1 oocyst per 10 litres. There was a full forensic chain of evidence, and there were very, very severe penalties imposed for failures or outbreaks. The response, the labs were forced to make significant investment in testing facilities. They also made significant investment in training and proficiency of analysts. But over time, the knowledge and the experience in the methodology increased significantly, and labs used to collaborate together to share knowledge and best practices, not just for performing the analysis itself, but for controlling Cryptosporidium. And over time, industry guidelines for water treatment improved. In the overall benefits, sanitary surveys of all catchments assess the risk of the pathogen being present in that influent water, that source water. The knowledge that utilities had with regards to their water system improved significantly. Water treatment improved along with infrastructure at plants that were deemed at risk. One additional benefit, the response to extreme weather events could be tailored to the catchment. So if there was a flooding event, they knew what to do to prevent an outbreak with Cryptosporidium. In general, overall water quality improved across the board. And that's not just for Cryptosporidium and Giardia, but also water treatment for your total coliforms, your E. coli, your enterococci improved significantly. Current status, the regulation was repealed and the onus was placed upon utilities to do their risk assessments. However, we expected the number of tests to drop, but the level of testing has been consistent. I think UK utilities see the benefits of it. Labs still test 1,000 litre drinking water samples where it's deemed necessary, so at risk sites, but they do far more raw water testing now to try and understand the water from where it's abstracted. And again, water quality continues to improve year on year. And with that, I thank you very, very much for your time. I hope that's been useful and insightful to you. And at that point, I will hand over to Aislinn.
Thank you very much. I was already introduced, but I am the area lead in the quality and process improvement team at Melbourne Water. And essentially my team provides process technical support to our field operators. And I'm just going to be taking you through our installation of Filta Max. Just to run you through what I'll be discussing today, I'll give you a little bit of a background of our Winnichy treatment plant. And then go through the monitoring that we have in place, the sort of reasons behind installing our Filta Max unit, which is largely related to our filtration system and good practice for filtration, and then take you through understanding risk and just our experience with the Filta Max installation. So Winnichy treatment plant is a conventional drinking water filtration plant. We have 5 clarifiers for sedimentation and 16 filters with sand media only. So it was built back in 1979, so it's a fairly old plant. It supplies about 25% of Melbourne's population with about 560 megalitres a day at the peak output. And it's supplied with water from Sugarloaf Reservoir. But Sugarloaf Reservoir is an off-stream storage, so it doesn't have its own catchment. It's supplied with water via pumping from 2 sources, the Yarra River and the Maroondah Aqueduct. The source water for this plant is classified as what we call Category 4 under the Australian Drinking Water Guidelines. So that means our catchment is unprotected. So there is industry, agriculture and recreation in the catchment. And the 95th percentile of historical E. coli data is in that range, 2,000 to 20,000 per 100 mils. So that is puts it in that Category 4 classification for those who are familiar with the Australian Drinking Water Guidelines. And Winnichy now includes a UV disinfection plant, which was recently built downstream of filtration, as the guidelines have updated regarding the required treatment and removal of protozoa in particular. So the monitoring required under the Australian Drinking Water Guidelines for a source for catchment. Monitoring for protozoa is on a risk-based frequency. So we're not given, it's not dictated how often we must monitor, but we must monitor based on the risk in the catchment. And typically our monitoring is carried out via grab sample, as most people are, I believe. So grab sampling is 10 litre samples compared to a plant throughput of 560 megalitres a day. So it really is a drop in the bucket or a needle in the haystack. So a very small volume to be assessing what our risk is at the plant. Filtration, as many of you probably already know, it operates of to media grains rather than size exclusion, as many particles are smaller than the pores between media grains. A key step in filtration as it removes the charge on particles which would prevent them from attaching to media. And turbidity is a measure of the success of removal of solids through filtration. However, turbidity has a number of limitations, including that Cryptosporidium oocysts are smaller than the wavelength of light used to measure turbidity. Also, a number of international studies have detected Cryptosporidium in filtered water at treatment plants, even when they were meeting turbidity targets. And internationally, there have been outbreaks of cryptosporidosis reported from areas supplied with water, even when they were meeting those turbidity targets. So there are certainly some limitations with the approach that is used to measure the effectiveness of filtration. So what we use instead is good practice. So as I said, there's evidence to suggest that turbidity can be relied upon as a measure of removal, but only when paired with good practice. And good practice, I mean, it's been defined in different ways across different countries and jurisdictions, but there are a number of excellent guides, including from the American Waterworks Association and Australian Water Association. So the good practice guides give recommendations for controls on water harvesting, filter design, operation of coagulation systems, backwashing of filters, monitoring and maximum flows through filters, and maximum rate of increase in flow. So I guess putting all this together, we wanted to understand more fully what was the risk facing our filters, and as I said, our sampling was previously infrequent, only weekly, and sample volumes are small, and so it provided only provided a real long-term view and a limited understanding of the risk facing our filters. So the Filta Max system, which we installed, I think, at the start of 2024, has allowed for much greater volumes of water to be analysed than what we had previously. So we are able to take, I think, we run water through it for 10 minutes every hour for a 24-hour period. And we do that 5 times a week. So much greater volume of water is processed through the filter. And this really makes it possible for us to actually make operational decisions around harvesting. So as I said, one of the key points around good practice is reducing your risk as much as possible through controlling your harvesting. So avoiding, I'll call it dirty water, or it's the higher risk water. And it allows us to modify our filter conditions to limit the maximum throughput through the filters when there is a higher risk, increase our backwash frequency, and reduce our filter run times when we do have that higher risk coming into the filters. So monitoring has been ongoing, and in recent times it has given us some really valuable data. I think we had, for a long period of the time that we had the Filta Max in place, we had quite a full reservoir, and we were diluting all our Yarra River water, which is the higher risk part of the catchment, with the cleaner, for want of a better word, Maroondah Aqueduct water, which is more of a protected catchment and has less risk sources. So we actually had very few to no detections for quite a long period. But in recent times, we have had some, I guess, lower rainfalls, as many people have probably experienced. And that means that our reservoir has dropped quite significantly, and we have had to pump water from the Yarra River without that dilution with our nice cleaner water. And that has been really interesting to see the results on the Filta Max, because we've actually started to get a few detections of Giardia, just impressing upon that this is still raw water. So a few detections of Giardia, and that's really verified for us the hydrodynamic modelling that we have done in the past that we sort of suspected was correct, but now we're able to actually verify that and build a picture of the risk profile to the filters. And I've just got a picture here on this slide of the installation of the Filta Max at Winnicki. It's installed in our lab. And it's been a really fantastic setup. I think the filter isn't actually in the rig at the moment. Unfortunately, I wasn't able to find a good photo, but it does show that it is a nice, neat little package, all just contained in that one area of our lab. So it has been a really useful tool, and we'll continue to use it to help gauge that risk to our filters and to help us control our harvesting to reduce risk as much as possible. So yeah, that's me, and thank you very much for your time.
Thank you so much, Aislinn, and thank you for joining us in this webinar. I'm going to drive a little bit of this question and answer session for you and for Brett. Well, thank you for all the insights on this. For sure, this webinar is going to be the first step to raise the questions, to bring up the conversation about the detection, but also the projects in Melbourne water. So for the questions and for everyone there, please, we have a tab dedicated just for the question and answer session. So if you have any questions, anything that you want to ask to Aislinn or to Brett, just click there and write your questions. We'll be happy to answer. If we cannot answer that questions in the meantime, or during this presentation, we will get back to you and we will reply with all the information that you're asking. So to start, I have a question for Aislinn. Are there any plans for Melbourne Water to roll out more detection systems at other treatment plants or points in the distribution network?
Many people might not be familiar with Melbourne's water supply system, but we actually are quite lucky in that a lot of our water comes from what we call protected catchments. So we don't have a risk of protozoa because they are closed to all human activity and all agriculture and all recreation. But we do have 3 catchments that are open or unprotected. So that's our Winnicky one, which I just discussed, Yan Yan and Tarago. And we do have tentative plans to include the Filta Max at Tarago and Yan Yan. But I will say that Winnicky is quite unique in the way that water is harvested into the reservoir, and the other 2 sites, it's actually understood because they have their own catchments. So we sort of, we're not kind of trying to test our different harvesting controls because, I mean, there aren't many. It's just a catchment and the water flows into the reservoir. So I think it would still be a valuable tool, and it still helps us to, as I said, control maximum filter rates and those kinds of run times and those kinds of things. But it's probably not as urgent as it was for Winnicky, for a better word.
Awesome. Thank you so much, Aislinn, and it will be nice to see the progress of your project too. I have a question for Brett here in the chat. Who installing a Filta Max based sampling system will reduce test cost for a utility? If so, in what ways?
Sorry, could you just repeat the question, David? I didn't catch the start.
Sure, definitely. Could you installing a Filta Max based sampling system help reduce the testing cost for a utility? If so, in what ways?
I don't think installing will reduce the costs for a utility. Certainly not the material costs. I think, however, by investing in the installation and the training to perform analysis for Cryptosporidium and or Giardia, in the long term, there are going to be fewer failures for those parameters, but also other parameters. And I think in the long term, there are going to be financial savings for that water utility. So it's very difficult to reduce the initial setup costs for the installation itself, for the training of the analysts, to get everything up to speed, to invest in the external quality assurance. But by doing that investment over a long-term period, you are going to reduce the cost because there will be fewer failures at water treatment plants. I hope that answered the question.
Oh, thank you. Thank you so much, Brett. I have here another question for Aislinn. Has the system revealed any unexpected trends or seasonal patterns in water quality that weren't visible with the Winnicky 10-liter sampling? So the actual model that is bigger, in terms of the sample, has this model revealed any seasonality or any seasonal patterns compared with the 10-liter model?
Yeah, I wouldn't say seasonal necessarily, but I think, as I said, when we had a nice full reservoir that was diluted with nice clean Maroondah aqueduct water, we didn't have any detections either on our grab sampling or on the Filta Max. But once we had that more empty reservoir and we started harvesting a bit more aggressively from the sort of dirtier Yarra River water, we started to get detections on the Filta Max, and we got nothing on the grub samples. So it actually means that we are getting data that we're missing through that grab sampling that we're doing. So it has been really valuable in that respect.
Okay. Could I just ask a question to Aislinn as well? I'm just interested to know, when you talk about water harvesting, I'm sure that's the same as abstraction. Where are you doing from that within the reservoir? Is it from the surface? Is it from certain locations?
Yes. So we do have a variable offtake our Sugarleaf Reservoir, but it hasn't been working for some time. So the offtake is actually quite low in the reservoir, I would say. So it's I'm sorry, I can't remember what the top water level is, but I think in terms of the full range of our offtake to our ability, we're taking quite low. And I think that's partly because in the late 2000s, so 2009, we had some drought conditions and we had quite a low reservoir, and we put our take depth very low, and that's where it stayed. So some works to rectify the variable offtake, but at the moment we're a bit restricted in terms of where we can take the water from.
Thank you so much, Aislinn. I have a question for Brett here. How well do the dyes distinguish dead from live protozoa?
So within current guidelines, such as US EPA method 1623.1 and ISO guidelines for testing, they don't distinguish. So the FITC stain will just bind to the outside of the oocyst or the cyst. The DAPI stain will just stain any nucleic acid, hopefully just within the nuclei of the Cryptosporidium and Giardia, but they don't distinguish necessarily between organisms which are alive or which are dead. There are some older techniques.
I also have a question that could be also connected to this. Oh, sorry.
Okay.
Go ahead.
So I was just saying there are some older techniques that have been used. One is called DAPI propidium iodide testing. So it was considered a viable organism. The organism was considered viable if it accepted the DAPI and excluded propidium iodide. So there are some techniques that you can do. But I'd also like to distinguish between what's considered a viable organism and an infective organism. And to know if it's infective, you can only do that with infectivity studies using cell culture or live animal infectivity studies. So it's quite complicated to do that.
Thank you, Red. As I was saying, we have another question connected to that one too, all in the same way. No way to detect troughs of Giardia?
A way to detect what of Giardia?
Troughs, protozoites. So is another station of in the Giardia cycle, if there is any way to not detect the cyst or all cyst, is to detect the
or the trophozoites. The trophozoites. Okay. Not really. Within the water system, what you're mainly going to be detecting is the cyst itself. So the infected host will shed millions of Giardia cysts, and you will be detecting those themselves. The only way to detect trophozoites, which by the way don't survive very long in the environment, would be to do some form of culture testing or fecal testing. to detect trophozoites. They don't survive long in the environment, so the reason for having the cyst is that it's more hardy in the environment and it can last longer until it finds its next host.
Awesome, thank you, Brett. Again, a question you will be able to answer. Is the initial filtration always done in water plant or 10 liter? Oh, well, you and probably John to this kind of question. And can 10 liter grab samples be filled? Oh, sorry, I lost. And 10 liter grab samples be filtered in the lab using the Filta Max. So can be done just in 10 liter and can be done 10 liter using Filta Max in the lab.
Yeah, so any water that's finished water testing will be done on site at a water treatment plant. So a drinking water treatment plant. That's done in a dedicated location using equipment very similar to what Aislinn is using now. 10 litre grabs do also come in, and it's more common now for 10 litre grab samples of the influent water to come into the laboratory, and they are still filtered in exactly the same way. We have sampling setups using peristaltic pumps or diaphragm pumps that we'll use to filter that water through the Filta Max system, and then you would go through exactly the same process. One other comment I would say here is that it's always a real balancing act with doing this. As Aislinn pointed out, you're taking a 10 litre grab from this huge reservoir. I can't remember how many megagallons it was, but it's a real snapshot. It's a drop in the ocean, really. But it's a real balancing act between trying to get what we would consider a representative sample volume, but also limiting the amount of particulate that you're getting in the sample that can affect some of the downstream procedures. And I think Aislinn has come up with, a great compromise with that by doing this periodic sampling over a longer period of time. So you're getting a more representative sample whilst reducing some of the interferences that you can get with the method.
Thank you, Brett. I have a very interesting question here for Aislinn. How do you decide the appropriate filter run times under dirty conditions as opposed to the normal conditions?
I guess we can see it through the head loss in the filter. So particularly when we have dirtier, and I want to put in inverted commas dirtier, because we actually, as I said, have had very few detections of protozoa, and our turbidity is, I think, like 2 NTU, which is pretty clean. But we can see the impact on head loss, and we don't really want to be getting closer to our terminal head loss where we're backwashing on head loss. So we prefer to be backwashing on a set time. And if we're starting to see that we're having to backwash the filters on head loss, then we want to bring that time back and actually backwash them on a frequency. It helps us to manage also our backwashing systems. So we don't want all our filters to kind of queue up at once, and then we've not got space in the wash water tanks. We want to be able to plan for it ahead. But I guess that high head loss condition is a risky one from a filtration perspective, because this sort of smaller spaces you have between your particles, the higher velocity your water is, and the more likely you are to shear off anything that you've attached to your media. So, as I said, that's one of the elements of good practice, because turbidity isn't necessarily a good measure of whether it's working or not. But you know that if you're consistently backwashing on high head loss, that you're probably at a higher risk scenario. I hope that answers the question.
Sure. Thank you so much, Aislinn. Question for Brett. What can you do to overcome troublesome samples containing interferences to obtain high recoveries? This question says, we have some samples that are not yielding good recoveries. So the question is how you can, in those problematic samples, how you improve the conditions to get better recoveries, Brett.
We have a phrase in the UK, how long is a piece of string? So it could be quite a long response. It depends on what the interference is. In some scenarios, particularly during the warmer months, you might start getting algal blooms. Sometimes they will be carried through some of the procedures, and it can make the identification of your target organisms much more difficult, particularly with use of DAPI stain, because the DAPI will stain all the nuclei within any algal cell, and you can often just get a flare and you really can't detect anything. Certain materials in water, if you've got any silicates in the water or siliceous materials, that could possibly affect the IMS step, can interfere with the IMS itself. High levels of particulates, so if you've got high levels of particularly ferric particulates, that can affect the IMS because it could be taken through the entire procedure itself. You can do additional wash steps to try and improve that. One thing that I would say that's probably very important is to use some form of internal control. So using something like colour seed, you can determine the overall recovery for a particular matrix, and then you can adjust your naturally occurring recoveries accordingly. So if I get 50% using colour seed and I get 20 naturally occurring oocysts, I can automatically adjust that to 40. So using an internal control such as colour seed is a really, really good idea, particularly if you do have those difficult matrices or certain seasonalities within your water system.
Thank you so much, Brett. We have a couple of questions, and given that we still have some time, I will try to ask them for you two guys. I have another question here for you, Brett. Can you comment on the potential for genotyping or of any detect crypto of Giardia?
Yeah. So genotyping is possible. It's quite a complicated process. What it involves is trying to extract organisms from a slide and then taking that through an extraction procedure and doing PCR on the sample itself. You could do it within your own laboratory if you have the expertise for PCR and the equipment for PCR. In the UK, what tends to happen is those samples where they believe they have positives is sent to a centralized expert laboratory that specializes in PCR for Cryptosporidium, Giardia, and other protozoa. So it's a complicated process, but there should be laboratories out there that can assist you with that.
Yeah. I have a last question for Aislinn. For Melbourne Water, what was the journey like from choosing the technology to actually installing the Filta Max based sampling rig? What were the key steps along the way?
Yeah, sure. Well, I guess we met with IDEX quite early on, so I think probably early 2023 we started having conversations about the technology, and I think it was my colleague Melissa who had experience with it in the past because she's worked in UK utilities, so she was familiar with them being used back when they were sort of part of the requirements. So yeah, we spoke to IDEX early on, and then we also spoke to some other utilities in Australia that are using the technology. So Sydney Water in particular were very helpful in giving us their experiences with the Filta Max. And then I guess the next step was also meeting with our lab provider to understand if they were able to process the samples. And originally I think they didn't have the elution station that's required for the processing, but we were able to resolve that and get them to come on board. Because I guess, it is a change to their usual method of analysis. So they were a little bit reluctant at first, but I think they saw the benefits and they were quite happy to include it. And then in terms of the actual installation, IDEX yourselves were quite helpful in recommending the control cabinet and the rig that you can see on my last slide there, which included all the controls and the electrics and the valves and the pumps and all the bits and pieces. So that all came in in one nice package. I think the only challenge we ran into is that because it was a UK company that installed that, it didn't meet some of Melbourne Water's very prescriptive standards, so we had a little bit of difficulty in getting it approved, but we did get it across the line, and actually it was exciting that our service provider that did the installation, when they got the rig, said, "Oh look, do this," and so I think IDEX potential conversations with them even to have sort of a local provider for those, and then so after that it was a matter of training up our operators to make sure they understood the operate the rig and the kind of process of changing in and out the filters, and that's gone from there. So.
Amazing, thank you, and thank you for the trust, Aislinn, and for participating in today's webinar. For sure, having this time for us is invaluable. We are almost out of time, so thank you, Aislinn, again, and Brett, for taking the time to share your expertise and your experiences with us in this excellent presentation. For everyone there, thank you for joining us. If you have more questions, and if you have any comments, please let us know, send us an email, and contact us. If you want to see this webinar on demand, for sure, we are going to have some links that we can share with you all. And if you have any other topics that we want to discuss, if you want a second part of this webinar, for sure, also let us know. Well, thank you, everyone, for joining us, and have a good day and a happy Friday. See you next time.